Hey, I'm Sam, and today I'd like to talk about a protein called CELF2 and its role in mRNA splicing. So let's start off by talking about pre-mRNA, the molecule that gets transcribed off of DNA inside of the cell nucleus. We all know the Lewis structure of mRNA. We know it, we're bachelor scholars. But we also know that we can write RNA as a sequence of nucleotide letters. But even this form of representing RNA isn't the most convenient, because for an entire mRNA, it's best to represent it as a diagram like this. Here you can see the main features of a pre-mRNA. You see the protein coding sequence, that's pretty self-explanatory. You see the 5' untranslated region and the 3' untranslated region. They don't code for protein, but they're still pretty important in the mRNA function. And finally, in a pre-mRNA, you will see introns. These are pretty long sequences of RNA that do not get transcribed, and in fact, they're often taken out of the pre-mRNA in a process called splicing. So splicing is a process that takes place inside the cell nucleus. In splicing, an intron might be removed from a pre-mRNA to form a mature mRNA without that intron, or perhaps it could be retained in the final mature mRNA product. Now you'll notice here that the intron that's retained, it occurs after the stop codon. So it certainly won't affect the sequence of the peptide that's translated, but it can still have important functional consequences for this mRNA. So splicing, you can see it looks pretty simple, but in fact it's extremely complex. And here you'll see the spliceosome, which is responsible for catalyzing the splicing reaction. The spliceosome is a huge mishmash of protein and mRNA, kind of like a ribosome, except the chemical reaction that it catalyzes is splicing as opposed to the lengthening of peptide chains. So the spliceosome has U1, 200 other RNAs and proteins, but even that isn't it. Outside of the spliceosome, there are over 100 splicing factors, such as CELF2, that help guide the spliceosome into making the right splicing choices. Now, you physics guys out there, I know what you're saying. You're saying, oh my god, 300 molecules? Why? It's so complicated, way too complicated. But really, we need those 300 molecules because splicing is hard. In splicing, the cell needs to achieve chemical specificity, sequence specificity, and regulation. Let's take chemical specificity. Well, the splicing reaction is really two transesterifications. Here's the first one, and to produce that product, and here's the second one. Now, wait, look here. See, in this transesterification, the 3' hydroxyl acts like a nucleophile, but the 2' does not. That's some remarkable chemical specificity orchestrated by the spliceosome. And finally, there's the product of splicing, with the intron taken out and eventually degraded. Now, if you look backwards a second, you might ask another question. How does the spliceosome know that this is a splice site? After all, it's just a sequence of nucleotide letters, and to the human eye, it's not too obvious where the exon ends and the intron begins. But to the spliceosome, it is quite obvious, because that, in red, is a splice site, and that is where splicing will occur. That's the sequence specificity of splicing. Finally, there's the regulation aspect of splicing. You can see here that this pre-mRNA, with its intron, tends to be spliced in a way in which the intron is removed. The bulk of it is flowing in this direction. But that won't always be the case. Perhaps a signal could stimulate the cell to change its mind, to start splicing this pre-mRNA in a way in which the intron is retained. And that regulation is a third challenge of splicing. So now that you have some appreciation for why splicing is hard, let's talk about the CELF2 protein, which is really the focus of my project. It's a fantastic protein. CELF2 protein has 3 RNA recognition motifs, and each one of these is pretty good at binding to mRNA and recognizing certain sequences inside of that mRNA. And you can see a structure of that recognition here. Now, one of the mRNAs, or the pre-mRNAs, that CELF2 protein binds and also influences the splicing of it influences the splicing of the CELF2 mRNA. And here's a picture of the CELF2 pre-mRNA right here. The first thing you should notice is that it's mostly intron. In fact, only about 0.5% of this pre-mRNA is coding sequence. And now look right here at the end, you'll see that the CELF2 pre-mRNA has a pretty substantial 3' UTR. And one thing that I have found is that the CELF2 protein is responsible for the alternative splicing of that 3' UTR. So you can see that there are two different potential 3' UTR products, and CELF2 protein is a deciding factor on which one is produced. Now you might be saying, look, Sam, I get there's intron retained in the 3' UTR, but really who cares? It doesn't influence the coding sequence. What's it matter? Well, hold on to that thought, because while it does not influence the peptide that this mRNA produces, it might have other effects on the mRNA. For instance, what if this process of CELF2 influencing the splicing of the CELF2 pre-mRNA leads to a more stable splice form, or maybe even a less stable splice form? This could greatly influence the future production of CELF2 protein in the cell. And my project, a big component of my project has been to figure out whether this is occurring. Does one of the splice isoforms have a more stable mRNA than the other? And well, how did I figure this out? I used a technique called quantitative PCR. Quantitative PCR, well, the first step is pretty easy. We're just going to take the mRNA out of the cell. Second step, also easy. Let's just reverse transcribe a single-stranded cDNA complementary to all of that mRNA. The third step is awesome. The third step, we're going to amplify this cDNA into double-stranded DNA. And as the double-stranded DNA forms, a molecule called cybergreen slips into the space in between the base pairs and fluoresces upon exposure to blue lights. So the fluorescence of cybergreen can be detected. That can quantify the amount of double-stranded DNA formed, and we can work backwards to figure out the amounts of each splice isoform of this mRNA. So that's pretty cool. This quantitative PCR has allowed us to quantify the amounts of each splice isoform inside of the cell. But really, when we think about it, that's not enough. I don't just want to know the amounts of the mRNA, I want to know the stability of each splice isoform. And to figure that out, I layer another method on top of quantitative PCR. And that's ethinyl pyridine labeling. So you can see here, this represents a cell, and each a happy cell, each triangle represents a piece of mRNA within that cell. So why don't we feed this cell a modified nucleobase called ethinyl uridine? You can see right here, it's not just a normal uridine because it possesses this chemical group, and will represent the mRNAs that contain ethinyl uridine as these red triangles. Now, let's just naturally go back to the regular uridine diet, and we'll see that the ethinyl uridine containing mRNAs, well, they stick around, but they kind of fade away. And because I can perform qPCR on just the mRNAs that contain the ethinyl uridine, I can determine the stability of each splice isoform of the CELF2 mRNA using quantitative PCR. So using quantitative PCR and this ethinyl uridine labeling, I've actually been able to show that the CELF2 protein influences the splicing of the CELF2 pre-mRNA in a way that produces a more or a less stable mRNA product. And this suggests that the CELF2 protein is acting in a way that will restrain the further production of CELF2 protein. That's pretty reasonable, right? Right now in my lab, and really for the past few weeks, I've been working really hard at figuring out how all of this occurs. How does CELF2 interact with the spliceosome to force the mRNA to be spliced in a certain way? And once the mRNA is formed, what makes it less stable than the alternative splice form? So those are questions that I continue to try to answer, and I've really enjoyed it so far. So I'd really like to thank my lab. These 4 people in particular have been fantastic, and I'm open to any questions you guys might have.
Sam Alon, the Science Mon.